So it ranges from kind of almost 0 to almost 1, and the average is a half, just not surprisingly. And moreover, the traits are, the heritability of the traits is not distributed at random. And in this example, I've just sort of highlighted skeletal traits, things like femur length or whatever, are about twice as heritable on average as reproductive traits. Traits sort of more closely aligned with fitness, we might think. So perhaps we're not surprised. The genetic variation for different traits is not random. Here is a slide from a great paper by Professor Poole, I see sitting in the back there. This is, so what he did here is plotted, basically this is the mutational variance, how much genetic variation is being input into the population by mutation, and this is the additive genetic variance in the population. This is 8 traits in Drosophila melanogaster, and we see this beautiful positive relationship with that sort of variance explained. So basically this, from this slide, you can say, okay, mutation is a very strong predictor of genetic variance in Drosophila. So another great paper, more recently, Brian Charlesworth wrote summarizing a lot of stuff, 50 years of data. I'm just going to give up on this pointer almost. Basically, it says there's too much genetic variation in traits related to fitness in Drosophila melanogaster to explain by mutation selection balance. There has to be some contribution from balancing selection. So I don't work on flies, I work on worms, and these are the traits that Risa and Dan looked at. These are 18 traits related to the first mitotic cell division, and things like the offset in the division plane, the elongation rate of the spindle, the initial length of the spindle, the final length. Some of these traits are reasonably highly correlated, a lot of them are not. So it's not much, we're looking at sort of all one big trait there. And Dave explained nicely what a mutation accumulation experiment is, and I probably don't have to explain to any of you guys, but for maybe one person who just wandered in, I will say that hope springs eternal here. So you start out with a highly genetically homogeneous population. You subdivide it into a bunch of independently evolving populations. You maintain them at very small effective population size, so selection is inefficient. You carry this out for a long time. Mutations accumulate independently in the different lines if you don't contaminate them. And then sort of at the end, you pull the ancestor, because the worms are cool, you can freeze them down, and then pull them out years later, and you can compare the mutated ancestor or mutated descendants to the putatively unmutated ancestor. And what you see typically is something like this, with the obligatory caveat, except in Arabidopsis, fitness declines because mutations are deleterious on average. You started out with no genetic variance in the population, and the genetic variance increases inexorably, and the slope of that line is the mutational variance, the end. So standing genetic variance, so this is the genetic variance that's present in the population. You go out and you look at a bunch of people like Polderman et al. Did, or flies like David did. So for a neutral trait, not under selection, at mutation drift equilibrium, the standing genetic variance, which I call VG, is proportional to effective population size times the mutational variance. So this is analogous to theta equals 4NE mu, right? For a selected trait at mutation selection balance, the ratio VG to VM is, let's say, the persistence time of a new mutation. Basically, this means how long does it take for your average deleterious mutation to get weeded out by selection. The flip side of that is the ratio VM, so the mutational variance, the standing genetic variance. This is approximately equal to the selection coefficient. So if the selection coefficient is 1, right, VM VG equals VM. Because the only genetic variance is the new mutations. Okay, so here's what we looked at. We had 2 sets of C. elegans mutation accumulation lines. They accumulated mutations for about 250 generations. So I started, so 2 starting genotypes, PB306, which is a wild-ish isolate, and 2, which is kind of the white lab mouse of C. elegans. And we looked at, or we, which is to say RISA, looked at 46 or 47 MA lines, manfully replicated the ancestor into what I call pseudo lines, and then lots of like 25x replication per line. So this is quite a highly replicated experiment. Then for the standing genetic variation, we looked at a collection of 97 wild isolates collected all over planet Earth. That was largely Eric Anderson's work. So here's the first set of results. So I've got the mutational variants plotted on the X. So the mutational variants measured in N2. So these are these 18 traits. So plotting mutational variants in N2 on the X and in PB306 on the Y. And this is the raw variance, but you see the same thing for if you standardize by the mean or the standard deviation or whatever. So basically this says that the mutational process is really consistent, at least with respect to this measure. So however a trait mutates in N2, it mutates approximately the same way in PB306, with this one sort of weird exception down there. Okay, so here's what, so you heard this first in School 98. So here we're plotting the standing genetic variants against the mutational variants, and VG almost perfectly predicts VM for these 18 traits. So the orange line there, that is the putative line of neutrality for any times VM using a sort of conservative but reasonable estimate of any for C. elegans based on nucleotide variation. So coming back to the Charlesworthian, what about balancing selection? So here I'm plotting VM on the X, and if life history, fitness-related traits have higher VM because they have a larger mutational target, then I think that's not an outrageous proposition. And somehow balancing selection is unique to this kind of trait, then what I expect is that if there's this additional little bump in genetic variation maintained by balancing selection for these traits, and these traits have higher VM, then if we plot VG against VM, we should see our high VM traits will be out here, and there should be this additional little bump up in genetic variation. So we have data on VG from several traits. So the blue, well, so we have a couple of different estimates of fitness. We have body size, and we have the red is another sort of life history-ish trait. And that's not what we see. We don't see this. It's not getting bumped up like that. These high VM traits are actually down farther below that line. So, at least to that extent, we don't see any evidence for that in C. elegans. Okay, so I guess it went really fast here, which is better than going really slow. So, the mutational process, at least with respect to these traits, is really repeatable. So I predict if I was to do this again in other C. elegans, I would see approximately the same thing. So for C. elegans, mutation is a sufficient and robust predictor of genetic variation. So we see a very, again, this has been seen in flies. It's also been seen in Mike Lynch's Daphnia lines, although those guys never analyzed the data in that exact way. And we see no signature balancing selection in this case. So I'll just end here and answer questions for a long time. And the person behind the next talk has been canceled. So Peter Lind will be giving the last talk in the session, so please don't abandon him. Come back in 20 minutes or so. Shad. Not for these traits. I desperately want Racer to do the same thing for Briggsy. So we've got estimates. So we have estimates of VG. We don't have the estimates of VM for Briggsy or for any of the other species.
So you either have VG or VM for the other species.
Yeah. No. So so we have some estimates of VG and VM for Briggsy, but not enough to do that kind of analysis.
So could it be that the lack of evidence for balance and selection is strictly due to the nature of phenotypes you're analyzing? Because if you're looking at repetitive output or other type of phenotypes, maybe you could see that.
I'm sorry, if I was looking at what?
Other types like repetitive output or other.
Yeah, so those were those traits we did look at that were for. Now the caveat that I didn't say is those data, the VG data for life history are mostly pretty crappy. And I would very much like to get better data for that. But as far as it goes, so now another possibility is that all the traits are if there was a consistent element of balancing selection for everything, right, then that would just sort of pick that slope up, right? And I think, I can't rule that out. David?
So are there mutational correlations among these first cell division traits that we would measure? Because that would sort of...
So, yes.
So, is it like...
So they're not super strong correlations, and they only they did this. We didn't do this for this full set. They were kind of adamant against wanting to do that. We did it for 6 traits. That was published in Current Biology. And there are some, so all of the traits they looked at are correlated more or less with embryo size. But the correlations aren't super strong. In particular, the phenotypic correlations are not super strong on average. Some of them are, but most of them aren't.